Good evening, students. This is our next video regarding megasporogenesis. In previous video, we studied about what is megasporogenesis, and here we will study about how your female gametophyte will become 7-celled 8-nucleate structure. Fine. So, monosporic embryo sac. Monosporic embryo sac is such kind of embryo sac where the development of embryo sac occurs from single megaspore, and it was studied by P. Maheshwari. But the explanation that in most of the cases of angiosperms, monosporic development occurs, and this monosporic development was first explained by Strasburger in Polygonum. Fine. So let's start. Here what happens once our single megaspore mother cell. Single megaspore mother cell is whom? How we studied about two cases. In case of crassinucleate ovule and in tenunucleate ovule. In case of crassinucleate ovule, archaeosporial cell undergoes mitotic division, single mitotic division, and form two cells, the primary parietal cell and the second is the primary sporogenous cell. In that case, sporogenous cell will undergo meiotic division to form 4 megaspores. And in case of tenunucleates, only archaeosporial cell behave like megaspore mother cell and it will give rise to formation of 4 megaspores. Here we are considering the monosporic embryo sac development, which our embryo sac will be having 7-celled and 8-nucleate structure. Fine. So we are having the single nuclei megaspore. Single cell megaspore कौन सा यहाँ पर functional होगा? The chalazal end. जो chalazal end पे present है, it will become functional and it will undergo 3 consecutive mitotic divisions which are free nuclear. मतलब यहाँ पर सिर्फ न्यूक्लियर डिवीजन होंगे, साइटोकाइनेसिस या सेल वॉल फॉर्मेशन बिल्कुल भी नहीं होगी, जिससे हमें क्या मिलेगा? एक 8 न्यूक्लियर स्टेज मिलेगी. दिस 8 न्यूक्लियर स्टेज में सेल वॉल फॉर्मेशन स्टार्ट होगी और वो अपने पोजीशंस को सेटल कर लेंगे और डेवलप हो जाएंगे इस पर्टिकुलर स्ट्रक्चर मेगा जो आपका ये मैच्योर एम्ब्रियो सैक है. ठीक है, तो यहाँ मैंने शो किया है माइक्रोपाइलर एंड पे. दिस इज आवर आर्केस्पोरियल सेल. जो आर्केस्पोरियल सेल है, जो यहाँ पर, या आप कह सकते हैं इसको प्राइमरी स्पोरोजिनस सेल, जो डेवलप होगा इनटू 4 मेगास्पोर्स. तो क्रासिन्यूक्लिएट में तो जो आर्केस्पोरियल है वो प्राइमरी स्पेराइटल और प्राइमरी स्पोरोजिनस सेल में ब्रेक होगा, जिसमें प्राइमरी स्पोरोजिनस सेल जो है वो मियोटिक डिवीजन करके ये 4 मेगास्पोर्स बनाएगा. और जिनके पास न्यूक्लियस पोरली डेवलप्ड होता है जैसे टेनिन्यूक्लिएट ओव्यूल, उनके केस में ये आर्केस्पोरियल सेल ही बिहेव करेगा एज मेगास्पोर मदर सेल. So this megaspore mother cell will undergo meiotic division and will form 4 megaspores. This is the micropylar end and this is the chalazal end. As I told before, the chalazal end megaspore will remain functional and rest of the 3 will get degenerated. The chalazal megaspore will undergo its nucleus, not the complete megaspore. The nucleus of the chalazal megaspore will undergo 3 consecutive mitotic divisions and it will become 8 nucleate structure. This 8 nucleate structure will they will form cell walls around them and they are localized at certain positions, forming this kind of mature embryo sac. So here it is the reverse portion, it is the chalazal end and this is the micropylar end. In the chalazal end, 3 of the cells will organize themselves as antipodal cells and these 3 cells are haploid in nature. Next, opposite to this, on micropylar end, egg apparatus is arranged. In this case of egg apparatus, 1 is the egg cell and 2 are the synergids, and all these 3 are haploid in nature. In center, 2 nuclei, they will not form any cell wall around them and behave as 2 polar nuclei inside a boundary called central cell. So here, if we start counting the number of cells here, the 3 are the antipodals, 3 cells, 3 are the egg apparatus, इनमें आपके 1 egg cell and 2 synergids. These are the total 6 cells and the 7th cell is the central cell. The 7 cells, but how many nucleus are there? Nucleus 1, 2, 3, 4, 5, 6, 7 and 8. So this is your 7-celled 8 nucleate structure. Let's discuss about their functions. So these 3 antipodals meant for providing nourishment to this central cell. Fine. Next, in case of egg apparatus, these two synergids are meant to nourish the egg cell. Fine. Here finger-like projections are shown. These finger-like projections are known as filiform apparatus. This filiform apparatus will guide the pollen tube to enter towards the micropylar end of the ovary. Fine. So this is our structure, the mature embryo sac, which developed from single megaspore, which was formed from nucellus. Fine. So this mature embryo sac is of 7 cells and 8 nucleus structure. This is very important structure. It is always asked in your board exams. So learn it very carefully, and each and every part of it and every function of each and every cell. Fine. In my next video, we will study about the how to calculate the number of meiotic and mitotic divisions in case of megaspores or megasporogenesis or in microsporogenesis. Such type of questions will be asked in your NEET exam as well as in your board exams. So be prepared for that. So we will study about the numericals again on the basis of microsporogenesis and megasporogenesis, including number of mitotic meiotic divisions if a particular seed is given, how many meiotic and mitotic divisions have taken place in the formation of that particular seed. Fine. So in my next video, we will study about the numericals, and this is all about the megasporogenesis. Thank you.